until he does the streaming. Let's see. Okay. Thanks a lot for the invitation and to the opportunity to talk about a bit about population dynamics in marine bacteria and what we can learn about that from gene expression analysis. Here is an example from a picture from our field station outside in the Baltic Sea, 10 kilometers offshore, that we sampled 2 weeks, every second week throughout the years since 2011. There is some nice colored image of a rhodopsin-containing bacterium that I will also talk about what are rhodopsins, to revive some history through that, because it seems to be some new people in the audience that I haven't seen before, so hopefully there will be something new. And for those of you who have seen some parts of it before, please have patience. I hope it will be something new, at least somewhere. My history is that I did my PhD at Umeå University in the north of Sweden. Then I went for a 1-year postdoc in Israel with Tom Beerman. And after that, I kind of reconsidered. Instead of going to the US, which everyone was doing at the time for a postdoc, I thought, well, I had a nice cruise together with most of the seniors here in Barcelona. So I thought, well, do the postdoc in Barcelona. I'll continue there. And stayed here for almost 4 years, working with Carlos Pedros, Celia, Pep, and Ramon, and several of the seniors. And then I went back to Sweden on a junior position in 2004, and since then I've also been on a sabbatical 2 years to the lab of Marianne Moran for diving a bit into transcriptomics and what that can help answering. So Kalmar is a beautiful place with a beautiful Renaissance castle. Our university maybe is not as beautiful, but we have people doing research on essentially the food web of organisms, plus some ecology people and birds and fish scientists. And if microbial ecology had been easy, it had been structured as it is here in this schedule. Unfortunately, it doesn't look like that in the sea, but it rather looks like this. This is a 5-millimeter tube, and maybe this is why you wanted me to talk again, just to see it. So this is just a tube put into seawater in the Baltic Sea and followed with dark field microscopy. And you can see how these organisms, they are not just floating around with inertia like we do when we swim. They decide precisely what they want to do and how to move. And so water at the scale of microbes is dense or with a high viscosity. So it's more like even copepods and even more the smaller organisms move like spiders on a wall. So when they move, they move. When they decide to stop, they instantly stop. I think that's quite clear from here. And the tiniest little dots that you can see in this picture are bacteria. And most of the bacteria in the sea, at least in the open ocean, don't move actively. They don't have flagella or anything. And here you can see how water is actually moving. Even as copepods move, the water moves like a gel at this scale. But these ciliates and stuff that circle around eat bacteria, and that's of course a fascinating subject of research that is upcoming here, I understand. And but then if we go back to time series analysis, you do this here too at Blanes Bay. And this is just a sketch of the temperature and some different images of things, major taxa that vary through the years. And I think we have very little understanding of what is it that makes some taxon, like the green here, which are cyanobacteria. Why are they abundant 1 year, or in relative abundance, high 1 year, whereas in other years, they are really few. But overall, they stay around, but there's still a fascinating variability in relative abundance of different taxa that I think we're far from understanding. And if we go down into the ASV level, or individual species or populations, we can see some taxa that are extremely, I mean, they come back every year at almost the same relative abundances, whereas we have other taxa that vary more, but essentially they return over time. And in the Baltic Sea, we have some upwelling events when we have big storms where the North Sea water comes into the Baltic Sea and push up deep water. That's when we can see deep water bacteria even in the surface. And that happens, it happens every year, but some years it's really a huge inflow that completely reshapes the water column. But I think I would like to understand why this variability between years and the amplitude of these peaks, what is it that regulates it? And of course, regulation is something awkward that we study in the first years of the biology courses or even biochemistry, where this central dogma, I think, is a bit underappreciated. Because we often look at it from the DNA to the RNA and the protein, and then we get frustrated when we see papers that the amount of DNA doesn't correspond to the RNA amounts that are produced, or even the protein and the activities. But maybe I think we should, from an ecological perspective, and even understanding, maybe it's more fruitful to look at it the other way around. To have a protein or an enzyme or a structural protein, we need to have had some sort of RNA produced in certain amounts that there is enough of the protein. And to have that expression of RNA, we need to have actually a certain amount of DNA in organisms of different types to actually ultimately generate the protein. So this concern about if there is a quantitative relationship between DNA, RNA, and protein, I think it's a misformulated question. It's rather that we need to have all three and to understand what gives us actually the functions, which is at the protein level in the end. Let's see if we can make, I don't have a very convincing case for that, but let's see. So if we look at 2 years at the LMO station, you can see some temperature, nitrate. We have typical seasonal variability across over 2 years. And if we measure gene expression analysis over this time, we can see that samples from June 1 year fall very close to June and the other year. And up here we have October 2016 and October 2017. It looks like, I mean, it matches what we see in 16S analysis, that things return, the same organisms more or less are there, but it means that they're also expressing similar things across the year, those taxa that return. So this is, of course, a nice figure. It shows something nice. The problem is that the underlying data is 300 different taxa. And going into the details and understanding the regulation of these, it's painful. But splitting it, maybe it helps a bit to split it up and show just easy examples of some pulse populations, some taxa that have quite short appearance in the water column, as detected by expression of nice, fairly short peaks, whereas other taxa have expression, they are there for much longer time, over 2, 3, even 5 months, with quite high expression levels. But ultimately, most of them, some of them even completely disappear from detection. So they're there and become abundant, but they're then essentially gone from detection level, for quite some time, but still they come back. Most of them, they are the same coming back. So to structure this even a little bit more, we can sort them into spring clusters or spring taxa, like we do in plants. I mean, we have spring flowers, summer flowers, and autumn. So then it becomes a bit easier for us to kind of look at the data and maybe try to sort out, do they all do the same or do they do different things? And if we take this kind of approach to just sorting them, I have shown you this before, we have a kind of a cascade of expression. This is, each line here is each row is a different species of heterotrophic or of bacteria, and each column is a date. So in winter, when we start sampling, we have some taxa. As spring comes, we get other taxa, and then we get into summer, other taxa, and then winter, and then it's winter, and then this taxon that was there in the first winter, comes back the second year, and then we get this cascade of expression of returning bacterial populations that do some sort of expression over time. So what do they actually do? And this essentially mirrors also 16S dynamics that we see over time and or maybe more used to seeing. So if we look into a taxon, an Aphanitsomina, it's a cyanobacterium, filamentous cyanobacterium that is forming kind of blooms, not toxic, but quite large summer blooms. An interesting thing here is that we have quite good detection of gene expression of this cyanobacterium throughout the year. So this, since it's there all the year, it can help us to tell what is it doing when it's not just blooming in summer, but at the rest of the year. And then we have here a cluster of genes that are highly expressed in summer, in the kind of peak periods. And then time goes, and then you have time on the x-axis. And then we have a whole bunch of genes, a broad diversity of genes in this genome that are expressed in the off-season. If we say off-season is non-summer seasons. And then the next summer, the same genes that were expressed the first summer turn back. And then when we come into autumn, October here, we're back to the set of genes being expressed that kind of indicate off-season. And if we sort these genes into functions, we can see that in summer we have usual suspects for a taxon that we know is nitrogen fixing. So they do nitrogen fixation, photosystems, especially pigments, are highly, highly expressed. But then in winter or in off seasons, we have more focus on cofactors, metabolisms included in maintenance to kind of survival mode over time. And then the summer transcripts come back again. So it's a kind of a nice positive control that seasonal analysis could tell us something. Because for this kind of organism, cyanobacteria, we know what they should do, but maybe not so much what they should do in the off-season. So I think it's... And if we look at then individual genes of this taxon, it's curious that we have some photosystem genes that are actually relatively higher expressed in the off-season, maybe providing the energy for adapting to slow growth conditions. Whereas, as I mentioned before, we have very high pigment expression. Nitrogen fixation is there only in summer, and it's essentially zero in the non-summer periods. So if we then turn to another heterotroph, and here we have no this here we don't know what they do. So here it's much more difficult to interpret. But this is the overall relative expression of a Pelagibacter, a SAR11 population. And it seems like, I mean, it's quite obvious that it grows from, in winter it increases, and then it has an increase period and a post-bloom period when it falls down again into autumn and winter. And in a similar way as the cyanobacterium, we here have, this is a bit difficult, but here we have the bloom period, but I would not have expected that the bloom period, I mean, I would have expected them to do the same expression pattern all over the period here. Because that's when they're kind of abundant, that's when they're happy. And at the lowest points, I would have expected them to have a different transcription pattern. But it actually seems that somewhere in the middle of the bloom, when they have grown, when they're on the falling out, that's when this shift occurs from the increase period, which is down here, and then they shift to another set of genes during the kind of post-bloom, but it's on the decay phase, or it's being outcompeted at this phase. And then the bloom genes turn back again this next period when this Pelagibacter is increasing. And we have some genes here associated with the post-bloom. Curiously, some vitamin genes. This is B2 and cobalamin B12 genes that are expressed. And that was lost. And in the bloom genes, we have ammonium uptake, we have thiamine genes, and something to deal with oxidative stress. So somehow we're sorting out that heterotrophs do adapt, but in ways that I wouldn't have directly expected. But this was a taxon, a Pelagibacter that is there for extended periods. It's several months that it's there. So it makes sense that over those several months, it actually shifts from one set of genes to another as being a focus. If we instead look at these short-term peak populations, which, I mean, short-term is relative, but I mean, these are 3-4 weeks of time when they are fairly abundant in the transcripts. And then it seems like this is a Pseudomonadales. Now almost all gamma protobacteria has been lumped into Pseudomonadales, so we have not been made a favor by the taxonomists, but I think we will adapt to that. I certainly think that the taxonomists are right on this, putting all things together, but it's a bit challenging. Anyway, here it's quite clear. If we look at a set of ecologically relevant genes of different sorts, everything from stress to phosphorus and stuff, I think it's quite clear that in the peak period, the 2 years, the relative abundance of several of those ecologically relevant genes is very similar. I mean, we have the same genes there and almost at the same proportions. So it seems like, at least for the pulse populations, we don't know when they are really low in transcripts, when they are out of season. Surely they can switch a lot, but it's curious that during the peak, they do precisely the same thing 2 years in a row. And that same taxon is on the left here, but if we then compare that one to a Roseobacter that also has 2 short peaks, the difference between different taxa is quite pronounced. So even though this Roseobacter also does the same thing here as it does in the same peak period next year, this one doesn't do the same as its co-fellow. And they are shifted a bit in when they occur in spring, and I don't know, but I would like to figure out if those genes contribute to decide precisely that pattern of that one in both years. I think the Rosibacter is coming after the Pseudomonas. But we're there. It's still a struggle to figure out. So from those kind of more general things, this is an example of focusing on the B1 thiamine expression, just to show an example of how the levels of expression can be a bit informative. So B1 is kind of a problematic vitamin both in the Baltic Sea and in some coastal areas and in the Great Lakes, because lack of B1 make salmon have a huge problem with their immune system. So they get infected, and it looks quite horrible. And it reduces the fertility tremendously. So the little fry, the eggs essentially die off if the mother or female hasn't had a good thiamine status during the previous months. And it's an open question, what causes this lack of thiamine? But the same thing, it also influences seabirds and gulls and ducks in similar way. They essentially get paralyzed and die. And with Ryan Pearl, he led a study that showed that this production of vitamins, of the thiamine, is largely regulated or encoded in heterotrophic bacteria and phytoplankton also, but that it varies a lot. The ability to synthesize this vitamin among microbes varies a lot between taxa. So we thought we should take a look at the expression of the genes involved to see if there are any patterns, which taxa actually synthesize. So this is which taxa actually synthesize thiamine and which taxa take it up from the environment. And like many vitamins, we know that B12, most of the vitamins we ourselves cannot synthesize them, right? So we're essentially relying on microbes to synthesize or plants to synthesize the vitamins. And in the case of B1, it's a very simple metabolic pathway to synthesize it. It's two branches that coincide and to form the final thiamine. But essentially there are different genes involved in the two branches and also in the uptake into the cell. This is a bacterial cell, also different genes for the uptake of the thiamine. And if we look at the expression over time of these genes, we can visualize it. We can see here that in summer, the expression patterns for these genes in thiamine synthesis differs compared to autumn and summer and winter. And if we look at it in relative expression levels across the community, we see that the community as a whole synthesizes more thiamine or expresses more genes for thiamine metabolism in summer. So these periods here with elevated values, that's for synthesis, and this is for transport into the cell. We can see it's lower relative transcription in winter and higher in summer when there is more activity. But still, I mean, even in winter, microbes need to do something, but then it shows that the relative levels are lower they do something else than thiamine synthesis in those times. And if one looks at the genome level or at the 16S, like 16S data, then one would be able to say that, yes, in relation to the number of cells, it's likely that this or that is happening. But if we look at metagenome level, then Well, in the upper left corner, you see there is a lot of green there. So in the synthesis way of part of the metabolism, it's all green there, and it's because cyanobacteria are really important and have very high expression levels of a part of the synthesis pathway. Whereas the purple on the left there, that's essentially alpha proteobacteria and a lot of pelagibacters, SAR11 taxa again, that do certain proportions of the synthesis. But the problem here is, or the good thing is, that it informs that on the expression of these genes, and actually the expression of the cyanobacteria is way much higher than their relative abundance in the metagenome or at the DNA level, because they are very highly, when the cells are expressed a lot in proportion to their biomass. So what we can get from the gene expression is the variety of taxa contributing to different processes. This is the thiamine uptake of a complete thiamine. There are many taxa doing it. Synthesis of the complete thiamine is pelagibacters and cyanobacteria. And then the very large dominance of cyanobacteria in different parts and dominance of pelagibacters. And if we would have averaged this or tried to restructure this from the metagenomics data, it would have looked much more even across all taxa. And yes, so in that way, this is just to say that different genes are expressed at different levels in the proportions that they have, and sorting out who express those genes kind of tell us in relation to the population dynamics what is going on. So with that, I thought of switching last to rhodopsins, and I'm not sure all know that rhodopsins, we have them in our eyes. That's the proteins that actually, where the photons hit our retina and send a signal to our brain that we see. So the rhodopsins are made for vision, but in bacteria, rhodopsins are used to generate energy, ATP. And they were discovered in the ocean around year 2000, and and since then it's been suggested that it's a kind of metabolism that we don't know how much energy, solar energy, that it actually captures. But we know that a grand majority of microbes encode the gene for the rhodopsin, and some bacteria use rhodopsins to survive better. So in this case, we show here that survival in the light is higher than in the dark. And other bacteria use rhodopsins to grow. So the growth curves in white are those in the light, whereas in the dark, those taxa, if there is low carbon, they don't grow. And the challenge is, okay, so they get some sort of energy. They get energy from this, from the rhodopsins, but which metabolisms is this energy actually channeled through? So to look at that, it's good to come back to the concept of the dogma in molecular biology from DNA to RNA. So this is just a growth of a Vibrio species as an example, where you can follow the optical density. So this is a growth. So all the genes that this organism have, of course, at the genome level, increases in abundance in proportion to its abundance. So all the genes in the genome will be there. But this is the expression of the rhodopsin gene. which is completely different, right? Because the expression here, when they're growing exponentially, they have organic carbon. They absolutely don't need additional energy. But the expression of this gene peaks at some point into stationary phase and then fades. But here it's important to remember that it's not that the rhodopsin disappears. I mean, we're used to things going away as disappearing, but this is the RNA. But the protein itself, the rhodopsins, that remains in the cells. So what you produced in this peak actually stays on. And this is what I think we're not used to thinking that something is produced and then it actually stays there until it's degraded. And of course, in the sea, we don't have these typical growth phases, so it's a bit more challenging to interpret natural data. And this is another experiment that we did with good organic matter yeast extract, as compared to challenging with only alanine, and we see that the rhodopsin stimulates more when having a bit more challenging source like as alanine. But light induces higher growth, but how? So then we did some expression analysis, and we could see, as we've seen before, that in the light, these are light, so the peak of expression is in the light, very low levels of expression in the dark of this flower bacterium. And then let's take another gene that is kind of completely unknown to most of you. It's isocitrate lyase. And if you look at the y-axis here, it's essentially 0 in the good substrate with yeast extract. It's almost not expressed, but it's extremely high relative expression in the light in the challenging substrate. So what is this weird isocitrate lyase doing? Well, it's linked with another malate synthase gene, which does the same, and this is a horrible picture to most of you, and I hated it throughout my most adult life. But this is the TCA cycle, you recognize it. What we mostly forget, at least I, is that this is where the carbon dioxide that we breathe, that we release, this is where it's produced. So this is important. I mean, we release, oxygen is needed for something completely different, but here we produce CO2. And the point in us and most heterotrophs is that when we release this CO2, that's when we get NADPH. Okay, so now it's getting biochemistry, it's annoying, but this NADH or NADPH is what goes through the electron transport chain where we get a huge amount of ATP. So if we wouldn't release CO2, we don't get ATP. But at the same time, if you're in a carbon-limited environment, it's a tremendous loss to lose that CO2. The carbon in the CO2 is just wasted, right? It's wasted because we need to get NADH to get ATP. But what these bacteria with the rhodopsin gene can do is that they can skip that step because they don't need ATP, because they have it from the rhodopsin. So they can activate isocitrate lyase and malate synthase and get succinate and get some NADH here anyway, but they can just save up on carbon. So I think this is what we think is a huge advantage and a metabolic way to use the energy to kind of save up on carbon. And that was in an isolate in a bottle in Kalmar. Who knows what's the truth of that? But this is from the Pacific, and where Otto Senetal, Ed DeLong's team, measured, first of all, light. This is light intensity over 3 days. The rhodopsin gene didn't vary in expression in the field in the Pacific so much, but the same gene as we found most responsive in our data set with our isolate is the one that has the most pronounced daily fluctuation in relation to light expression. So it seems like isocitrate lyase is really a marker gene for photoheterotrophy using rhodopsins. And then the long team went on and they have done another time series over 3 days, and where you can see that the expression of this isocitrate DAG is just tremendously beautifully linked with light and this dial changes in growth conditions. And so now we're precisely doing experiments in Vigo, in Galicia, out here at Illa de la Toralla, so close to that ugly high building and next to the beautiful luxury houses. There is their ESIMAT, which is their marine field station at the University of Vigo, and we're now doing experiments to look at these sets of gene expressions. We're using this, I think this is our material standing here. And sampling many bottles. Sandra Martinez Garcia is our host, and it's really simple-minded things, putting seawater into bags, which they do, unfortunately not bottles, but and in this case we're adding different sorts of substrates, primarily different protein and amino acids, to see how they differ. So just as a take-home, I think it's important to remember that any process rates, or many process rates that we measure as a bulk entity, depend on both the community turnover, that is the changes in population dynamics, but also the regulation of gene expression at certain levels. And then the linkage between DNA, RNA, and protein is what actually is the basis for this kind of, for the changes in the processes that we measure. And to be useful to society, except for understanding more, which is kind of nice and useful, I think we have a task to do something like the hobby ornithologist society has done, or a community has done. And this is this wonderful breeding atlas of Europe's birds, linked with habitat distribution and distribution, to be able to say, this species exists here, this is a very widespread, beautiful little woodpecker, but we have taxa that are really limited in space, right, and are really endangered, and that depend on certain habitat. And I'm quite convinced that we could and should provide kind of a similar atlas of our coastal seas to tell where along our coasts do we need certain microbiomes, partners, to help processing what we throw into the water and help fisheries and whatever. But I think we have some homework to do to make ourselves understood as useful in that context as the ornithologists have done. So with that, I thank the funders and you for listening. And there are some opportunities, so if you know someone who is interested in a PhD or a postdoc, don't hesitate to contact me. Thanks. Any questions?
Questions?
Thanks, Aaron, for the interesting talk. I was wondering if you think it's necessary to have a good knowledge of the presence of these microorganisms in the system before doing transcriptomics, or you think, or maybe switching to quantitative transcriptomics, or I don't know which, because it also, as you see in the cultures, you see there's a huge change, right, in the through the growth curve.
Yeah.
And I mean, even so, we always do transcriptomics, and it's relative abundance of transcripts, but maybe they are still there or are not expressing some genes, or maybe they are just not there. So which one would you think will be if you had a lot of money and time and hands, which one would be the best approach to know really what's happening?
Yeah. I mean, I think those of us who have worked with 16S data, we're so used to relative abundance data. So I'm kind of happy with that. So I think relative expression data, it tells, just as we interpret relative abundance of taxa from 16S data, and we never question whether those are, well, Ramon questions whether those are real or not. You're the only one that has expressed it. And that's a good argument, and the main argument for doing fish analysis with specific probes to really, because, I mean, how can we really know they exist? And Ramon, the answer back then was we need to do fish probes to quantify things. So, and then you get absolute data.
No, I don't mean you cannot get information from it. Of course you can, but I mean, now we're trying to move actually to quantitative 16S data with standard, and so you have there's a way to do quantitative transcriptomics as well.
Yes.
But would you, so if you had a lot of money, would you sample both metatranscriptomes and metagenomes, or would you just stick to metatranscriptomes?
I mean, the metagenomes cost much less, and that's, I think, why people have been doing it more.
Yeah.
But metagenomes are quite expensive too, so I don't really know why. I don't have it. I think the methodology is there to interpret. I think of gene expression as a response variable. So when you push the system a bit with something, with a nutrient or with a temperature, you get a change in growth conditions, and that's when you see a shift in the allocation of RNA to a certain process. It's more challenging in a stable system where they just do business as usual. Well.
Okay, thanks.
Yeah, related to Marta's question, thank you very much, because I'm one of the new people seeing this, so I find your results super, super interesting in terms of ecology, theoretical ecology, and understanding how you showed first the drop, and you showed that the microorganisms are moving wherever they want and doing whatever they want to do. And then I think that Marta's question, for me, if I have a lot of money, I think transcriptomics is the best, because then you know what they are doing. And then with the metabarcoding, you can know why they are doing so, related to the community composition. So if you repeat this experiment in several years, maybe you can see why they are blooming, they are doing something, they change to do something else, and why sometimes they do that, and in another bloom they don't do that. And I'm convinced it's because of the community composition, who is there and what they are doing. And maybe you can disentangle all this with transcriptomics.
And I personally like a lot 16S analysis. I love it because I love different species, if it's birds or bacteria, but I think it tells us a lot about, I mean, that's when we go out in the forest. We interpret what, with different plants flowering or growing at different times. We don't need to do the biochemistry of that plant to know that it's there and flowering in spring. So I think just seeing something associated with the time is very helpful. Because, yeah.
Very nice talk, Jeroen. And I found interesting your take of the dogma of the relationship between the three pools of DNA, RNA, and protein, hence function eventually. What maybe you forgot, or you did not mention, is another aspect, which is another aspect that regulates this relationship or has a very important impact on this relationship, which is the turnover of each of the pools, and especially the RNA and protein. And you kind of said, oh, the protein persists for hours, days, months. Is that true? Or, I mean, if it's short-lived things,
yes,
they don't stay in the cell. They need to be produced all the time, and that's the holy grail to understand seasonal cycles, but also diel cycles and very short reactions or responses to stimulus and things like that.
And I think one of the easiest Nature papers that any of you could do, and maybe someone is doing, or I should have done it 15 years ago, is to do this thing and just measure. I mean, here is a tremendous amplitude in the expression of the isocitrate lyase. But I'm sure that the pulse of this is enormous. But the abundance of the isocitrate lyase enzyme, I'm convinced it's stable. Because it does, but to keep it stable, you have this pulse of production every day. Do you see what I mean? So I haven't. It's just that's how I view things. I mean, the protein. There is no chance that the protein is going up and down like this. But the production that we see, this diel variation in bacterial production, it's usually much more higher amplitude than the abundance of cells. And that's the same thing. I mean, to maintain the population of SAR11s, you have these peaks in activity. Just like we don't eat at night. I mean, but we're not disappearing at night, right? Humans. So microbes take things in periods. And we see that with bacterial production, and we see it with activity measures over day and the DMSP turnover, right? But I'm convinced that the proteins for that are there stably. But to keep it stable, that's my take on it. That then we get these pulses in the transcript.
Who knows?
It's a piece of cake to do the paper.
No. I mean, it's totally reasonable what you're saying, but then the derivative of what you're saying is that the activity, the final function, is kept kind of invariant. And when you have the chance, the very few chances where you can measure the activity together with that, you see that there is pulses or cycles or whatever in the activity as well, meaning that the protein is acting also in pulses or probably it all depends on what we're talking about, or which one.
Yes.
It will change from one to another, but so still to interpret how expression relates to activity, that's still very challenging because we don't know whether it's that the expression is just to keep proteins high, as you say, or there at enough levels, or it's related to the activities. And if you look at the Rubisco, you see that the expression goes up and down, the gene expression. I don't know about the protein, but I guess that the protein works with light, and I don't know. I mean, it all depends on the turnover.
No, and this is what I think in that Nature paper that any of us should do. There will be measures of the production of the protein coupled with the functioning of that of whatever enzyme or some DMSP lyase or something. And then, but not only that, but also the feeding of substrate into that process. So in your case, it would be DMSP, I suppose, because the DMSP you have shown varies tremendously, and that's a phytoplankton production in most cases. No, so it's the rate of a process depends both on our favorite microbes, but also on the substrates and for a certain process. But I'm intrigued. That's just so this stable maintenance of the protein. That's just for me, because I can't. I mean, this doesn't exist on the cell level. I mean, maybe there is a bit of change, but that's fine. But there is no way the cell levels of SAR11 are shifting 25 times. It doesn't happen on a diel cycle. So it just makes life easy. I figure the proteins are there, but to keep processes possible to carry out, we need these pulses in activity of many genes. Not all genes, because not all genes are responsible. This is the rhodopsin gene. This is the, or now I don't remember if it's the rhodopsin. The gray thing there is maybe the rhodopsin gene. I need to check it. But you see, that one is stable, right? Fairly stable, as compared to, so not all things in a cell varies 25 times over the diel cycle.
Yeah, thanks, Jerome, for this talk. Yeah. Thank you very much. It was extremely interesting. You are presenting this hypothesis that the pulses of expression and then the constancy of the protein. I also doubt about that, because really then it is what is the meaning, no? And the activity, we know that there are pulses. So I think you should make this Nature paper to measure, for instance, in this case, how this enzyme during the samples.
Yeah,
do you know how to do it? Or?
Yes, because well, I haven't thought about it. Well, I've thought about it, but you can quantify the end products. So with metabolomics, you can quantify how much succinate you produce, how much glyoxylate, and how much malate, and the resource coming in. So one can quantify it, I think.
And then perhaps the amount of enzyme, it's directly related to the function, or could be that the enzyme is there and it's not working at that particular moment?
I mean, if there is no substrate for it, it won't work. I mean, if there is no isocitrate, in this case, just as theoretical, if there is no isocitrate, there will be no activity of this one. So there needs to be something feeding into the system. So like, I mean, you need to, the proteins need to eat something for the degradation of that thing to start.
And just to finish, I like a lot the fact that you have a reference genome, and then when you pump the activity in different moments, different genes were being expressed by the same species at different times. This differential expression, which is something that I wouldn't have predicted, in fact. I don't know how common was that, but my question is what is the representation of this set of differential expressed genes to the complete gene expression? Was a significant part, a small or a large?
It varies over the season. It can vary from 15% up to 55%. I think 55 is maybe the largest proportion we have.
Which is still
So
significant.
it's quite some, no, it's not 5%. No,
Any other questions? Oh.
Follow-up. Because
there is no younger one that dares to.
That's a follow-up of this one.
Yeah, sorry.
Can the cell maintain its isocitrate lyase, because I also didn't listen at the biochemistry classes. I hate them.
I didn't even pass them.
Okay.
I didn't do them.
But I thought that these enzymes are expensive to simply maintain them.
Yes.
So if there is no isocitrate, then the enzyme
Exactly.
degrades.
So, and that is why, in this case, in the case of alanine, you have extremely high expression of these two enzymes. But in yeast extract, it's almost zero. So it's extremely strongly regulated by the substrate availability and the quality of the substrate. So if they have yeast extract is really wonderful food. That's the beef. It is literally beef. So then they don't spend energy on doing those two enzymes if they have resources available. There are many there are many considerations here why I say alanine is bad food. It's just because it contains 3 carbon atoms. So when you run it through here, and you lose one carbon and another one there is only one carbon left. And then you cannot, then it's impossible to continue the TCA cycle. So there was a trick, and that was, we actually figured it out before the experiment. So alanine is impossible to use if you cannot kind of activate these enzymes. But we never guessed that it would activate them more than any other gene. So the substrate quality, the quality of food is critical here.
Thank you for your talk. I wanted to ask about different things. I like the cascade thing that you showed. That was very interesting. I liked it a lot. But I wanted to ask on the expression of, it was Pelagibacter, that was this bacteria that was the whole year. And you showed all expression of different genes during bloom and post-bloom phases. Did you expected those genes in each phase, or what did you expect?
No, I had no clue.
You had
I mean, I would have guessed that during that entire period, maybe the guess would be that it would be during the entire period when transcription is high there in the left panel, they would do the same thing because they would be growing happily, and even the decay phase there, I would say maybe it's flagellate grazing catching up with them or something. I wouldn't have expected, so I would have expected maybe, of course, ribosomal proteins and TCA cycle enzymes, but no, this is just a selection, so it's not fair to all of you. But it pops up larger differences between the bloom period, I mean the increase period and the decrease period. I didn't expect that at all. Mm-hmm. And we know very little about what heterotrophic bacteria do in general, so I haven't had many expectations in beforehand.
Yeah. Okay.
But it seems that I was a bit surprised that post-bloom things, it seems to be geared towards maintenance, similar to in the cyanobacterium, where they try to kind of manage life in some sort. And it's also quite clear, not here so much, but the number of genes that, because this is relative expression data, the number of genes that are activated during the bloom period, when things are happy, you get it's like 16S data, you get a lower diversity when you have a strong bloom. It's because you have few taxa contributing to a bloom. It's the similar thing with the expression, that it's relative abundance data in transcription, so you have quite few genes that turn up when they are very, very active, because you get this dominance. The reads are stolen kind of and taken to those metabolisms that are very highly expressed. And then you get a much larger diversity of expression in off seasons, because they kind of even out. Well, it was a long answer to me. Some other question maybe.
Well, thank you very much. If there's no more questions, we can close this talk. And thank you very much, Jeroen.
Thank you.